Okay, so the next thing in our little video that we're going to talk about is membranes and how important membranes are for cell biology. So, cell membranes, membrane proteins, methods to study, membranes and their associated proteins. This is all going to be covered in this chapter. So, let's first talk about membrane structure. So, membranes all are composed of the same material, but it's just in different ratios. They have an amphipathic or amphiphilic structure, which is called the phospholipid bilayer. And what that basically is it has the two fatty acid tails on every phospholipid bilayer, and these tails, so one is solid. So these tails, they form spontaneously in solution. So these phospholipids will spontaneously form lipid bilayers in aqueous environments, which is really, really important for you to know and understand that whenever there's an aqueous environment, these bilayer structures will be formed. And these bilayer structures have proteins embedded into it. So that's why they're so important when it comes to, later on protein synthesis and all of that. So with phospholipids, with their structure specifically, it has one hydrocarbon tail that is unsaturated, and the unsaturated one is the one with the kink in it, and it allows it to move. So that's that. So length is important for specialized membrane structures to form, and phase transition we have liquid to crystalline depending on temperatures and lipids. So when we talk about phase transition, obviously solid is saturated and liquid is unsaturated. So the main thing that are involved in phospholipid bilayers is hydrophobic force and van der Waals interactions, and that's what's important in determining their structure. Okay, that was a lot of information I just threw at you. Let's slow it a little bit down and talk about the major flavors of phospholipids. So we have phosphatidylcholine, phosphatidylphenylamine, phosphatidylserine, and sphingomyelin. These are different phospholipids, and you're just going to learn about these later on and try to understand them a little bit better. So that's that. And we're also going to talk about, phosphatidylcholine and sphingomyelin. So phosphatidylcholine, that is going to be PC, and sphingomyelin is SM. PC and SM, these are normally on the outside of the plasma membrane. So they're usually sugars and they're on the outside of the plasma membrane. So they're on the outside. And then we have phosphatidylserine and phosphatidylphenylamine, and they're on the inside of the plasma membrane. So phosphatidylserine, which is PS, and phosphatidylphenylamine, which is PE. These are on the inside of the plasma membrane. So these different kinds of phospholipids is important for you to understand because these are specific kind of lipids. They're called glycolipids. So glycolipids are sugars that are outside on the cell, and remember we talked about that in carbs where they can modify different carbs to glycolipids and all of that. So that's very important for you to know. So we talked about the structure and all of that, and we're also going to talk about entropy and free energy. So entropy means disorder, and spontaneity means free energy. So when the spontaneousness goes down, there's more order. But when the spontaneous goes up, there's more disorder. So just remember that, in future things. And let's move on to the next thing that we're going to talk about, and which is cholesterol. So cholesterol, you have to remember that cholesterol is very, very strong in structure. It's in all eukaryotic membranes. So remember that it's something that's just exclusive for eukaryotes, and it's not in prokaryotes. And it's structurally a little bit more rigid than phospholipids. So it has a very rigid look. So it makes these cholesterol. So just think about cholesterol, rigid, really strong. So they make this cholesterol makes the membranes a lot less fluid because of its rigidity. And when the cellular membrane is a lot less fluid, it decreases the permeability to let small water soluble molecules to let in. So it makes the membrane a lot more strong. So it prevents hydrocarbon chains from coming together and crystallizing. So once you have that cholesterol stiffened area, the region of fluidity is a lot less. So just remember that for the future. So they're a lot more. Just remember for cholesterol that they're a lot more rigid than phospholipids, and they can't really rotate, and all of that. So the more cholesterol you have, the less you lose fluidity. So just remember that for cholesterol and all of that. So the next thing we're going to talk about is lipid rafts, and that's coming up next. So what are lipid rafts? So basically lipid rafts are a little bit more saturated hydrocarbons. So they have more sphingolipids and cholesterol. So what's that? So they have a little bit longer and straighter lipid tails. So these lipid rafts have a little bit longer, straighter tails like this. So just remember that. So they have the unsaturated hydrocarbon chains, they have the saturated hydrocarbon chains. So that's basically what it is. So a normal phospholipid has a saturated and an unsaturated section, right? But in lipid rafts, it's all saturated hydrocarbon chains. And in a lipid raft, you find more saturated fatty acid tails and more SM and cholesterol. So that's the main thing. So in lipid raft, there's more SM, which is sphingomyelin, and cholesterol. So just remember that. And remember, cholesterol lets there be less fluidity than all of that. So when we talk about lipid rafts, so when it's unsaturated, it keeps our membranes fluid. Always remember that. When things are unsaturated, it keeps our membranes fluid. So when they're saturated, they become a little bit stronger. That's why there's cholesterol inside of them. So just remember that. And also when lipid rafts, they're more saturated, so they're longer and straighter lipid tails, and do have particular proteins and all of that going on. So once you remove that, you can understand it a little bit better. So we talked about lipid rafts, and we're just gonna go a little bit into more detail about what lipid rafts are. And they're basically act as signaling platforms in cells. So they do create structures, and certain proteins are involved in transducing, signal localizing to these domains, and which is really, really important. So these lipid rafts specialize in signal transduction, and that is regulated in immune systems by lipid rafts. So one of the main things that lipid rafts do is they let signal transduction happen, and that's regulated in the immune system with the help of cholesterol. So biochemically, these domains can be distinguished from the rest of the plasma membranes, and we have learned that if we remove cholesterol with beta-methylcyclodextrin, this disturbs the structure of the lipid rafts, and in result we lose signal transduction of some receptors, particularly in the immune receptors. So that's how we learn that these lipid rafts specialize in signal transduction in the immune system. Is we learned that once we take out the cholesterol with the beta-methylcyclodextrin, it really disturbs the structure, and there is a huge loss of signal transduction in the receptors in the immune system. So there's a lot of asymmetry in phospholipids in membranes too, and you don't need to remember a lot of that, but there's a lot of flip-flopping going on where when one membrane is made of one cell or when the cell is distressed. So there's apoptosis and all of that. So you know that apoptosis is when a cell dies, and glycolipids always on the non-cytosolic side of the membrane due to how they are made. You will understand this later. So when I talked about how I believe SM and PC, I think, were on the outside. These are glycolipids, so they can only be on the non-cytosolic side of the membrane, and this is due to how they are made and how they can't really enter the cytosol. So that's that. Now we're going to talk about something very, very important, which is called transmembrane proteins. We're going to go in a little bit more depth, but basically what transmembrane proteins are is they're kind of in the membrane a little bit, and they're amphipathic, and they're meant for modification. So there's many different kinds of transmembrane proteins. There's the alpha helix is a transmembrane segment of a protein. Beta sheets, they can be integral membrane proteins. So you got beta barrels, you got single pass. Single pass is when it goes through once, and multi-pass proteins is when obviously it goes through many times, and you're going to learn more about that later. Okay, bye.